Okay, well, it's a real pleasure to be here. Thanks for inviting me. I'm kind of going to go to the opposite end of the spectrum from people that you've heard today, in that we're trying to build something that's really simple and will be very cheap and will work in places like sub-Saharan Africa, where there's heat, humidity, little or no stable power, and relatively unskilled personnel, and lots of dust. So high tech, we're thinking, is not going to be quite so easy to use in those kinds of places. So really we're focused on the fact that personalized medicine is almost certainly going to be the medical care of the future. And currently, medical care is basically one size fits all. So depending on what you've got, there's a standard treatment, and regardless of your genetic composition or that of your disease, you're going to get pretty much the standard treatment. But things are changing, and the system is going to, which is the, pointer?
The one here. The pointer is in the middle.
Oh, okay. Thank you. So it's basically tailoring treatment to the molecular characteristics of the person, because as the host, our bodies determine how we respond to drugs, and of the disease, because the molecular characteristics of the disease determine how that disease will respond to particular drugs. And so this is actually a really sensible strategy, because it saves the people from having to endure bad side effects from treatments that won't help them, and it helps the healthcare system, because expensive treatments are not being used for people who will not respond. And in order to implement that, we really need a way to have affordable medical testing. And a really critical issue here is the need to test patients one at a time. To the extent that molecular medicine is being practiced now, it's usually done in the context of batch testing. So many samples are collected at a central reference point and tested when there's enough samples to run the test. So you might need 100, you might need 1,000, depending on the test, which means there are delays shipping things back and forth, and you're not getting a result immediately at the time the clinician is likely to want to try to initiate treatment. So a one-at-a-time device is really a critical issue for efficient molecular medicine. And in addition, usually we're going to need results from more than one test. Now, you heard multiplexing mentioned earlier today. I think it was in Matthew's talk. Multiplexing is doing many molecular reactions in one reaction vessel. It's very complex. It's a difficult balancing act. It frequently doesn't work. It's probably not what you want to be doing in, let's say, sub-Saharan Africa. So we've gone for something we call multi-parameter, which is still doing multiple tests simultaneously, so a whole panel at the same time, but each in a different reaction vessel. And I think our approach, which I'm going to tell you about, makes all of this feasible. So in many respects, we're responding to a market pull rather than a technology push, in that we know there's a need for molecular testing and a need for an affordable way to carry it out in the clinic at the time the patient is in the clinic. In other words, at the point of care. So we've used miniaturization, and although our team has the word microfluidics in it, we've actually moved away from microfluidics. We may very well be using it at some point in sample delivery, but what I'll tell you about really does not involve microfluidics. But we have small sample volume, fast processing. We're going towards portable instrumentation. We're not there yet. It'll be low cost and automated, which means the user simply need add the sample, press go, and a result will come out. The user doesn't actually look at some of the molecular data that I'll show you, just so you can see what this can do. So it'll be easy to use in resource-poor areas, but also in community hospitals. I live out past Stony Plain, and they're not doing molecular testing out there. So it'd be a way of being able to get testing to people close to where they live, even in rural environments. And it'll actually save time and money in sophisticated urban hospitals as well. And it'll probably change the way clinical trials are done because of the ease of molecular testing. And importantly, it'll be feasible in extreme environments. So here's our device. I've got one here if anybody wants to look at it. It's basically just a polymer, essentially like Jell-O, that's put into a mold with all the reagents needed for a molecular reaction. I'll tell you a little bit about the molecular reactions in a minute. And then it's peeled out, and each of these individual, what we call gel posts, and here they are, they're a millimeter by a millimeter. Here's an array of an 8 by 8 array, or I guess it's a 9 by 9 array. It could be any configuration. One makes the mold to fit the use that's needed. peeled off, and we're going to have to enclose it into a cassette kind of structure, and we're working on that now. So that the user only needs to add the sample. All the reagents are here, and they're polymerized within the gel. So there's no need for pumps and valves. There's no need for reagent delivery. Just show someone, here's the mold and the final device being put. At the moment, we just have a little black pan that sits on a device I'll show you in a minute, which is a really simple device. But clearly, to be used anywhere other than in our lab, this needs to be enclosed, and we have some really, I think, innovative new approaches for doing this. So here's our current device. It's really, really, really low tech. It's basically an old-fashioned ring stand. Here's a Peltier, which is a heating element. It's the little black pan with the gel post array is sitting inside that. The molecular reactions need heating and cooling. So the Peltier can heat very rapidly and very efficiently and very precisely. And we've developed a lot of really standardized procedures to know, with multiple different of these instruments, to know that we're actually reaching the appropriate temperatures, because that's a pretty important issue. And also it addresses the issue of standardization. But cooling can be slow, so it's got a heat sink, which is this fan of copper stuff. And then the laser that excites, all the measurements are done using a fluorescent dye that can be excited under particular conditions. And the laser is a Blu-ray laser from a DVD player. It's cheap. It's under $50. The camera, which records the image, is a deep sky imager that's an amateur type of camera that, again, is quite inexpensive. So we're really developing this off-the-shelf instrumentation, which is still coming. And even with off-the-shelf things and building something identically, we're finding with multiple instruments, they aren't the same, which I guess is predictable. And so standardizing is critical, and we're now learning where the variability occurs, which is going to be really important for commercialization. I keep forgetting I have to push something over there. So our readout for all of the tests is going to be laser-induced emission of light, or fluorescence, as the method of detection. So here's our mold, here's the gel post, and this is just a mock-up where every post has been given the conditions where it will fluoresce appropriately. And you can see how the green dye, and so we can take a picture of that. We can actually take a picture of that over time, as I'll show you, to get a quantitative measure of the number of genetic copies of something that was originally present. And the dye emits light only if the reaction is positive, and all tests use the same dye. So there's no need for multiple colors, multiple lasers, complex optical detection techniques, which again is a big advantage. Having to have multiple colors in a given reaction adds enormous complexity that we wish to avoid, and actually are able successfully to avoid. So this little cartoon just shows the process of polymerase chain reaction. So that when we look at genes, molecular biologists can't really effectively look at one copy of a gene. But we can look at billions or millions or billions of copies. So this process is basically a molecular Xerox machine. So the DNA is, by adjusting the temperature, the DNA is denatured, so you can, it opens up. Here it's opened up. And a copying mechanism comes in, copies, makes a copy, then it's closed up again, then it's opened again, more copying. So by about 35 cycles of this molecular copying, you have 68 billion copies. Well, you can do a lot with 68 billion copies. Most importantly for us, you can detect them very effectively on these gel post arrays. So polymerase chain reaction is just one of the types of methods that we can use on this, but it's a very effective one for being able to do molecular testing. So here's an example of the results. So here's our device. It'll eventually look something like this, where everything's compressed. We haven't taken the time to do that yet. And so we've got all the multiple posts. And here we're actually looking, so dye is also polymerized in these posts. Sample's been added, and the reaction started by the heating cycling conditions. And at the beginning, the first few cycles, there's no detectable product because it's really too low. But then there's a period where you get a logarithmic increase in the amount of fluorescence because there's more double-stranded DNA because of the molecular copying, and then it levels off. So if you do a yes-no test, you're usually measuring up here, which means you could have 1,000 times more original copies of the gene in your reaction, and you get exactly the same result as if you only had 100 or 1,000-fold less copies.